Welcome back to our channel Ignited Medicos. This is Rachana. In the previous video, we have learned about transamination, and in this video, we shall be continuing with deamination and metabolism of ammonia under the subheadings formation, transport and storage, functions, disposal and toxicity of ammonia. Deamination. It is a removal of alpha amino group from the amino acid. This is the basic structure of amino acid. This is carboxyl end, also called C-terminal, and this is N-terminal, amine end. R represents the side chain. The carbon which lies next to carboxyl carbon is called alpha carbon. The removal of alpha amino group from the amino acid is called deamination, which is catalyzed by the enzymes deaminases, which forms corresponding keto acids, and free ammonia is liberated. High ammonia levels in the blood is toxic, which will be discussed in the next topic. And carbon skeleton is involved in different pathway and can be utilized for energy production. It occurs primarily in liver and kidney, mostly in the liver, and to some extent it occurs in kidney. There are two types of deamination: oxidative deamination and non-oxidative deamination. Oxidative deamination. Diamynation reaction is coupled with oxidation. Oxidation is nothing but removal of hydrogens from the amino acid, and deamination is nothing but removal of amino group from the amino acid. So it means there is removal of hydrogens along with the removal of amino group. This reaction is catalyzed by the enzyme Glutamate Dehydrogenase, which requires the coenzyme NAD or NADP. Significance of oxidative deamination. It mainly takes place in the liver, and to some extent it occurs in the kidneys. It provides nitrogen in the form of ammonia for urea synthesis. Amino group of most of the amino acid is transferred to alpha ketoglutarate to produce glutamate. Glutamate is oxidatively deaminated by glutamate dehydrogenase. This enzyme is present only in the mitochondria of liver. And glutamate dehydrogenase can utilize NAD+ or NADP as coenzyme. Non-oxidative deamination. There is removal of amino group without oxidation. So there is no removal of hydrogen from the amino acid, only amino group is removed from the amino acid. Enzymes involved are amino acid dehydrases. These are PLP-dependent dehydrases. These enzymes act on hydroxy group containing amino acids and form a respective alpha keto acid with the liberation of ammonia. Amino acid desulfohydrases are the enzymes which acts on sulfur containing amino acids. These undergo deamination coupled with desulfohydration to give keto acid pyruvate. Deamination of histidine. The enzyme histidase acts on amino acid histidine to liberate ammonia and by the non-oxidative deamination. So our next topic is metabolism of ammonia. First we'll look at formation of ammonia. The major source of ammonia in our body is by transamination and deamination reactions. And small amount of ammonia is formed by catabolism of purines and pyrimidines. Also, ammonia is formed from biogenic amines. In intestine, by the action of intestinal bacteria on urea, ammonia is liberated. And in kidneys, it is used for the maintenance of extracellular pH. Ammonia can easily be diffused across the membranes, but at physiological pH, ammonia exists as ammonium ion. This ammonium ion is hydrophilic, so it is not freely diffusible. In the next topic, we'll look why ammonia is highly toxic to central nervous system, and detoxification of ammonia is by conversion to urea and excretion through urine. Coming to our next topic, transport and storage. In most of the tissues and muscles, proteins undergo proteolysis to form amino acids. This occurs when there is decreased glucose level in the blood during starvation and fasting, which causes a decrease in insulin and increase in glucagon hormones, which stimulate gluconeogenesis. Gluconeogenesis is nothing but formation of glucose from non-carbohydrate source, which bring back normal glucose levels in the blood. So this is a skeletal muscle. Most of the proteins are present in skeletal muscles and they undergo proteolysis to form amino acids. These amino acids undergo transamination reaction with alpha ketoglutarate to form glutamate. So here the carbon skeleton of amino acid is converted to alpha keto acid, which involves in different pathway for production of energy. Now the amino group which is transferred to the keto acid forming amino acid glutamate. This glutamate undergo transamination reaction with the pyruvate catalyzed by the enzyme alanine aminotransferase ALT, which forms alanine and alpha ketoglutarate. This pyruvate is the end product of glycolysis. The amino group from the amino acid is transferred to the glutamate first and then to the alanine. Now this alanine moves into the blood from the skeletal muscle and reaches the liver. In the liver, alanine undergoes transamination reaction which is reversibly catalyzed by the enzyme ALT. Alanine is converted into pyruvate and alpha ketoglutarate is converted into glutamate. When in the stressful condition like starvation and fasting, pyruvate is converted into glucose through gluconeogenesis. This glucose is released into blood, which is transported to the brain cells, neurons, and to the RBCs primarily because they need the most, and some amount is taken up by the skeletal muscle and undergo glycolysis to form pyruvate, and this pyruvate will further undergo transamination reaction and form glucose-alanine cycle. Now if we look here, this amino group of amino acid in the skeletal muscle is transferred to the glutamate of the liver. Now this glutamate can undergo glutamate dehydrogenase reaction, which is broken down into alpha ketoglutarate and ammonia is liberated. This ammonia enters the urea cycle and is excreted through urine. Similarly, in other tissues also, proteins undergo proteolysis to form amino acids. These amino acids undergo transamination reaction to form glutamate. Now, ammonia is added to glutamate, which is catalyzed by the enzyme glutamine synthase and forms glutamine. This reaction requires energy, and the glutamine which is formed is transported to liver, intestine, and kidney. In the liver, glutamine is catalyzed by the enzyme glutaminase to form glutamate and ammonia. This ammonia gets involved in urea cycle and excreted through urine. Functions of ammonia. Ammonia is a toxic nitrogenous product, but it has various functions in our body. It is involved in synthesis of purines and pyrimidines. It is also involved in synthesis of non-essential amino acids like asparagine. Ammonium ions are very important to maintain acid-base balance in our body. Bicarbonate and ammonia acts as chemical buffer to maintain blood pH. Also, they are involved in synthesis of amino sugars like glycosaminoglycans. Disposal of ammonia. Based on type of nitrogenous waste excreted, organisms are classified into three types. Aminotelic, in which the aquatic animals excrete ammonia directly into water. Uricotelic, ammonia is converted into uric acid and then excreted, as in reptiles and birds. Ureotelic organisms, mammals convert ammonia to urea, and urea is non-toxic and soluble compound, so it is easily excreted. Ammonia from all over body reaches liver, wherein it is detoxified by the liver cells and excreted through the kidney. Urea is osmotically active, which means it would cause osmosis, and high concentration in plasma can affect osmolality. Urea is ideal nitrogenous excretory product as it is highly soluble in water and large amount can be easily be excreted, and it is non-toxic and metabolically inert. Toxicity of ammonia. Normally, liver has to eliminate ammonia through urea cycle. When formation of ammonia is more than capacity of liver to eliminate, then it may lead to ammonia toxicity. Or in the case of liver diseases like hepatic cirrhosis, ammonia gets built up when liver can't eliminate it properly. If ammonia is accumulated more than the threshold, then the reactions are reversed. Ammonia reacts with alpha ketoglutarate to form glutamate, which is catalyzed by the same enzyme glutamate dehydrogenase. And to this glutamate, one more ammonium ion is added to form glutamine, catalyzed by glutamine synthase enzyme. Since the concentration of ammonia is high, it utilizes more alpha ketoglutarate to convert it into glutamate. So the concentration of alpha ketoglutarate decreases. Alpha ketoglutarate is an intermediate of Krebs cycle. So there is decrease in there will be decrease in production of ATP, which directly affects the central nervous system. Symptoms include slurring of speech, blurring of vision, nausea and vomiting, and neurological manifestation, convulsions.